Alright, back to this overview of fat and carbohydrate metabolism. We have now covered all the pathways that I wanted to go through. For carbohydrates, these pathways included glycolysis, homolactic fermentation, the pyruvate dehydrogenase complex, the citric acid cycle, the electron transport chain, gluconeogenesis, and lastly, the synthesis and breakdown of glycogen. For fats, the catabolic pathways were the breakdown of triacylglycerol and beta oxidation, which tied back to the citric acid cycle and the electron transport chain. On the anabolic side, we covered ketogenesis and the synthesis of fatty acids and triacylglycerols. You can see on the left the metabolic cell and the extent of all the reactions that are diagrammed on the right. It goes without saying that there are a few concepts that I left out of our discussion, namely the metabolism of other lipids like phospholipids and cholesterol, or the metabolism of amino acids and nucleic acids, which tie pretty neatly into this picture. Some of these concepts are already integrated in the model cell, and when the time comes, I will make videos on these topics to understand how they relate to fats and carbohydrates. Now, to finish our discussion on the metabolism of fats and carbs, I want to cover the hormonal control in more detail. As of yet, if we consider some of the pathways where hormones were important for regulation, I have left out many details such as where the hormones come from and by what mechanism they operate. I simply discussed at what step they perform their action and whether it had a stimulatory or inhibitory effect. Additionally, if we set aside the reactions for a minute and zoom on top of the metabolic cell, I have yet to put into perspective the overall effects of a given hormone on fat and carbohydrate metabolism. In this diagram, the green and red arrows correspond to stimulations and inhibitions respectively. As you can see, all the hormones have multiple targets across the metabolic cell. As such, there will be three goals to this section. First, understand where the hormones come from and the conditions that promote their release. Then, understand what their enzymatic cascades look like. And finally, understand their global effects on fat and carbohydrate metabolism. Now, given the crucial importance of fat and carbohydrate metabolism to the cell, these pathways are used all across the body, and there is a wide variety of hormones that can influence those pathways. For the purposes of this video, the three main hormones that we will consider are insulin, epinephrine, and glucagon. These three hormones generally represent the main regulators of fat and carbohydrate metabolism, and for that reason, we will focus on them. To understand how these hormones are integrated in the human body, let's first go through an overview of the main organs that will be relevant in this discussion. This will allow us to get some intuition on the process and introduce a bunch of relevant terminology. To start, let's consider the organs that consume fat and carbs the most and for what purposes. In this category, we have muscles, adipose tissue, and the brain. To put it simply, adipose tissue are cells of fat, and their main function is to store the excess lipids and carbohydrates in the form of triacylglycerols all over the body. On the other hand, muscle use fatty acids, carbohydrates, and under certain conditions ketone bodies to generate energy as a source of fuel for muscle contractions. In terms of anabolism, muscles are capable of storing small reserves of glucose in the form of glycogen. When it comes to the brain, this organ is a little different from the other two, as it does not metabolize fatty acids. Instead, the brain runs pretty much exclusively on carbohydrates and in certain conditions, ketone bodies. All this energy is required by neurons to fuel their electrical activity and by glial cells to perform their functions in the nervous system. Now, given that the fats and carbohydrates that these organs use come from our diet, there are a few organs in the digestive system that are important to consider, namely the liver and the pancreas, which we can see behind the stomach and the intestines. If we first consider the pancreas, it generally has two functions. First, it helps the body digest by releasing enzymes that will degrade the food, and second, which is the function relevant to us, it releases hormones, namely insulin and glucagon. Insulin generally gets released after a meal and is typically seen as a signal that lets the rest of the body know that nutrients are present. As such, insulin decreases the levels of nutrients in the blood. Inversely, glucagon is released during periods of fasting, starvation, or exercise and contributes to increasing the nutrient levels in the blood. Now, when it comes to the liver, This is an organ that has so many functions that it's hard to summarize what it does in one sentence. Given its central role in the digestive system, the liver has numerous functions that are related in helping the body digest nutrients. For our purposes, what is important about the liver is that it constitutes the primary anabolic organ. What is meant by that is that the liver is sort of a central hub that makes sure all the other tissues across the body have all the nutrients they need. Accordingly, the liver is capable of performing gluconeogenesis, ketogenesis, storage and breakdown of fat and glycogen, and many more functions, all for the purposes of serving the rest of the body. Finally, the last organ that I want to consider is the kidney, which can be found behind all the organs of the digestive system. Although the kidneys play some metabolic roles that are similar to the liver, since their main task is to filter and clean the blood, the reason why this organ is important for us is because on top of them, we can find the adrenal glands. Out of the many things these glands can release, the adrenal glands can release epinephrine in conditions of stress and exercise. Together with the pancreas, this is where our hormones come from. From this overview, hopefully you can start getting an idea of how things are organized in the human body. Due to our multicellular organization, different organs are specialized for different tasks and thus need the appropriate signals to know what to do when food is present or not present. With this overview in mind, I want to start with insulin, which will give us a good foundation to understand glucagon and epinephrine. Accordingly, let's consider the pancreas to understand the release mechanism and downstream effects of insulin. As I mentioned previously, the pancreas has two main functions: help the body digest and secrete hormones. If we take a closer look at this organ, these two functions are mediated and separated from each other by different kinds of cells. The vast majority of the organ is made out of pancreatic acinar cells that perform the digestive function and connect back to the intestinal tract. The hormonal function can be located around these acinar cells in structures called islets of Langerhans. These islets contain a wide variety of cells capable of secreting different hormones. In the case of insulin, the specific cell type that is responsible for its production is called the beta cell. As a molecule, insulin is a peptide hormone, which simply means that it is a protein made from a gene. The exact mechanism of how insulin is produced is not super relevant for us. The only detail to consider is that insulin eventually gets packed into vesicles within the beta cells. When it comes to the release of insulin into the blood, the signal that triggers the release is food itself. More precisely, glucose is the main nutrient that is responsible for triggering the release. Now, although I won't go into as much detail for the other two hormones, I want to give some intuition on how glucose absorption after a meal is linked with insulin release. So, the first step in this process is mediated by glucose transporters, also called GLUT, and allow beta cells to absorb glucose. In the case of beta cells, GLUT2 is the transporter, and we will see shortly why the variant of GLUT is important to consider. The glucose that was just uptaken can then be processed by all the catabolic pathways we have covered, so glycolysis, the pyruvate dehydrogenase complex, the citric acid cycle, and the electron transport chain to produce ATP from the mitochondrion. The reason why ATP production is important is because beta cells express ATP-sensitive potassium channels. At rest, these channels remain open and continuously pump potassium outside the cell. Accordingly, with minimal glucose uptake, beta cells have a net negative charge inside. When ATP accumulates from the accumulation of glucose, ATP inhibits the potassium channel and forces potassium to stay inside, which depolarizes the cell. In other words, the charge now becomes more positive. This depolarization causes the opening of voltage-gated calcium channels and lets calcium enter the cell. Inside, calcium acts as a second messenger and has a multitude of targets. One of its targets is a protein located on the vesicles that links calcium entry with fusion of vesicles to the membrane. As such, the entry of calcium causes the exocytosis of the vesicles and by association, the release of insulin into the bloodstream. You can see that insulin release in the blood is ultimately linked with how much glucose there is in the blood to begin with. When there is high blood glucose, which happens after a meal, insulin release will be promoted, but when levels are low, for example during starvation, the release will be prevented. Although we won't dive too deep into this aspect, I want to mention that there are a few ways to modulate the release of insulin. To name a few, incretins, cholecystokinin or CCK, the presence of certain amino acids like arginine and leucine, as well as the stimulation of beta cells via the parasympathetic nervous system through acetylcholine, all increase the release of insulin. On the other hand, stimulation from the sympathetic nervous system, which operates through norepinephrine, inhibits the release of insulin. With this being said, now that we have insulin in the blood, let's see what happens when the hormone reaches its targets. Based on the organs that I introduced before, insulin mostly has a great deal of influence on the metabolism of muscle cells, adipose tissue, also known as adipocytes, and cells of the liver called hepatocytes. Accordingly, all these cell types express the receptor for insulin. To avoid repeating information across all these cell types, I will diagram the effects of insulin in one cell. As you will see shortly, the different tissues respond differently to insulin, and I'll make the difference explicit on the right. For that reason, the diagram on the left will not fit either tissue. It will simply summarize all the effects insulin has. Alright, before we get into the effects of insulin, we first have to consider what class of receptor it even activates and what cascade of proteins that triggers. So, the insulin receptor is also known as a receptor tyrosine kinase, or simply RTK. This receptor type generally has two important sites: an external ligand binding domain that binds insulin, and an internal kinase domain with tyrosine residues. When insulin is bound and the receptor gets activated, it subsequently activates the kinase domain and induces an autophosphorylation of its tyrosine residues. These phosphorylated residues can then attract additional proteins called adapter proteins that link the receptor to a wide variety of other enzymes and pathways. In the context of metabolism, one important adapter protein it attracts is IRS1. IRS1 allows for a kinase called PI3K to bind. This kinase is a dimer of two subunits, P85 and P110, and as you can see, P85 allows the linkage with IRS1. On the other hand, P110 is the kinase domain, and its main action is to phosphorylate the phospholipid PIP2 and turn it into PIP3. PIP3 can then activate two important kinases: protein kinase B, which is also known as AKT, and PDK1. PDK1 can phosphorylate PKB and activate it even further. In this entire pathway, PKB is really what opens the door for insulin to have its effects, as it has a ton of targets that it can phosphorylate, which means it has a ton of influence on various pathways. As we will see later, the analogous protein to PKB for glucagon and epinephrine is pKa. As a quick note, since enzymatic pathways are always a work in progress, and I do not want to make this video extremely heavy on the protein names, I will spare the details of how PKB performs its downstream functions. If we now consider the effects of insulin on metabolism, the only direct target of PKB that I want to mention is the protein AS160, which is an important regulator for GLUT4 insertion. As you might recognize in the name, GLUT4 is a particular class of glucose transporters. These transporters are very interesting because they essentially stay dormant in the cell until an insulin signal causes their translocation to the membrane. With more glucose transporters, cells can take up more glucose from the blood. In terms of our tissue of interest, only adipocytes and muscle cells are responsive to insulin in this manner. The liver does not express GLUT4. Nonetheless, the liver will still absorb glucose after meals as it expresses GLUT2 like the pancreas. The GLUT2 transporter, however, is not sensitive to insulin. Now, with glucose inside the cell, insulin can do a bunch of different things depending on the tissue. If we first consider the muscles, the main impact insulin will be the synthesis of glycogen from all the extra glucose that was just absorbed from the blood. More precisely, insulin will inhibit GSK3 beta and stimulate PP1, which altogether will promote the activity of glycogen synthase, thus promoting glycogen synthesis. If ever the proteins that I mentioned when describing how a certain hormone impacts a pathway seem foreign, I suggest going back to the respective sections of the pathways and taking a look at the regulation mechanisms to get a reminder. For example, with glycogen, the regulation pathway looks like this, and these diagrams should be helpful to understand the context behind the proteins that I am talking about. This applies to all the subsequent effects I will describe with the hormones. This way, the video can focus more on integrating everything together rather than jumping back into past topics. With this being said, if we consider how adipocytes react to insulin, instead of storing their glucose in the form of glycogen, adipocytes will store their excess glucose in the form of triacylglycerols. Recall that the two ingredients to make triacylglycerols are fatty acids and glycerol molecules. These two components can be obtained from glucose, as glycolysis links to the production of glycerol, and the degradation of pyruvate through the PDC produces acetyl-CoA. Acetyl-CoA can then be used to synthesize fatty acids. All four of these pathways, glycolysis, the PDC, fatty acid synthesis, and triacylglycerol synthesis, get stimulated by insulin. For glycolysis, insulin stimulates PFK2, which results in the stimulation of PFK1 and thus forces glucose to be degraded into glycerol and pyruvate. As a quick note, all the other sugars that we covered, so fructose, galactose, and mannose, can contribute to this pathway. For the PDC, insulin stimulates the pyruvate dehydrogenase phosphatase, which activates the complex and causes the production of more acetyl-CoAs. In fatty acid synthesis, insulin stimulates the enzyme ACC and accordingly promotes fatty acid synthesis by increasing the concentration of malonyl-CoAs. Since malonyl-CoA is the main inhibitor of beta oxidation, insulin also indirectly prevents the degradation of fatty acids. Finally, insulin promotes triacylglycerol synthesis by its inhibition of HSL, which prevents the degradation of those triacylglycerols. When it comes to the liver, hepatocytes respond to insulin by both increasing their stores in glycogen and triacylglycerols, so they perform all the pathways that muscle cells and adipocytes do. Hopefully, you can see that with respect to fat and carbohydrate metabolism, insulin is simply a hormone that signals to the rest of the body that nutrients are present and tissues should feed on them as much as possible to replenish their stores. Ultimately, this allows to decrease the levels of nutrients in the blood. If we consider this diagram that we have been working with since the start of this discussion, the net effect of insulin looks a little bit like this. All the nutrients get shunted towards anabolism. Now, unfortunately, insulin is not that simple. It turns out that it is a much more complex hormone than what is shown here, as due to its RTK receptor, insulin can also link to a mitogenic pathway that has a wide variety of effects in the nucleus. One of these pathways is called the MAPK pathway and reunites all these proteins to ultimately reach the DNA. Inside the nucleus, the pathway can impact gene expression, cell growth, and many cell differentiation pathways, among other things. The details of this pathway are not too important for us. The takeaway message here is that insulin is very complex and has a ton of downstream effects in the cell. This simple diagram that is shown here does not scratch the surface of what it truly does. Finally, to end this segment on insulin, I want to briefly discuss how the signal can be terminated. For this, there are two main proteins that mediate this role. First, there is PTEN, which is a phosphatase that converts PIP3 back to PIP2 and thus shuts down PKB and PDK1. Second, there is PTP1B, which is also a phosphatase, but in this case dephosphorylates the tyrosine residue of the receptor, leading to the termination of the signal. This last detail concludes what I wanted to say about insulin. Now back to this diagram, let's now explore the effects of glucagon by considering the pancreas again. Glucagon, just like insulin, is a peptide hormone produced in the islets of Langerhans, but instead of being produced in beta cells, glucagon actually gets manufactured in alpha cells. The main physiological signals that promote glucagon release are low levels of glucose in the blood, also known as hypoglycemia, and exercise. The mechanism that links blood glucose levels and the release of glucagon is pretty similar to what happens in beta cells, with potassium and calcium channels being involved. However, the dynamics are flipped. In the case of alpha cells, the intuition is that at low levels of glucose, the electrical activity of the cell favors calcium entry and thus exocytosis. But at high levels of glucose, calcium channels close and prevent the release of glucagon. If you want more information on the electrophysiology of alpha and by association beta cells, I highly suggest consulting this article that reviews how those mechanisms are possible. For our discussion, the details are not super important other than knowing that low glucose promotes glucagon release. Also, before we move on, I want to mention that glucagon release is stimulated by epinephrine and inhibited by insulin. When we will get to epinephrine signaling later on, you will see why it's important to keep that detail in mind. Now, when it comes to the targets of glucagon, The main target that we will consider in this video is the liver. As such, we can directly assume the cell to be a hepatocyte. In reality, glucagon receptors are more widespread around the body, including adipocytes, but since the effects of glucagon there are the same as those of epinephrine, I'll simply focus on what happens in the liver. So, based on what we saw previously with the different organs, the purpose of glucagon is to signal to the liver that there is a lack of nutrients and something should be done to make sure the rest of the body has all the appropriate levels of energy. In the case of starvation, some of the main tissues at risk of being nutrient deprived are the brain and the muscles, including the heart. The two main resources that the tissues can consume to keep working are glucose and ketone bodies, which can both get generated in the liver and be distributed across the body. To understand how this happens, we first need to discuss what type of receptor the glucagon receptor is. So, it turns out that the glucagon receptor is a G-protein coupled receptor that is associated with the GS pathway. In this pathway, the binding of glucagon activates the receptor and causes it to then activate a G protein by exchanging the GDP of the alpha subunit to a GTP. This activation frees the alpha subunit and leads it to activate another protein by the name of adenylyl cyclase. When active, adenylyl cyclase is responsible for converting ATP molecules to cyclic AMP or simply cAMP. This cAMP can then activate protein kinase A, also known as pKa. As I said during our discussion on insulin, pKa is the key enzyme that can now phosphorylate a bunch of substrates and regulate a wide variety of pathways. With respect to the metabolism of fats and carbs in the liver, all the pathways that pKa regulates from glucagon are catered to remake glucose and ketone bodies such that they can be released in the blood. If we first consider the glucose side, it can be made by two general pathways: the degradation of glycogen or via gluconeogenesis. In the context of glycogen, pKa acts on four targets. It stimulates the PP1 inhibitor as well as the phosphorylase kinase, and it inhibits PP1 and glycogen synthase. Altogether, these regulations favor the breakdown of glycogen to get back some glucose. In terms of gluconeogenesis, glucagon inhibits both PFK2 and pyruvate kinase, which shuts down glycolysis and thus allows glucose to be remade. Besides pyruvate, the initial substrates for gluconeogenesis can also be lactate, intermediates of the citric acid cycle, and a select few amino acids. Now, when it comes to ketogenesis, glucagon indirectly stimulates this pathway by increasing the concentration of acetyl-CoAs. To do so, the hormone promotes both the breakdown of triacylglycerols by stimulating HSL and ATGL, as well as the breakdown of fatty acids by inhibiting ACC. With reduced ACC activity, it allows to decrease the concentrations of malonyl-CoA and thus remove the brakes on beta oxidation. Beyond making ketone bodies, fat catabolism in the liver is super important because it supplies the energy required to perform gluconeogenesis. Recall that gluconeogenesis is an anabolic process, and so it requires some energy to occur. Altogether, the effect of glucagon on the liver look like this when we consider this pathway diagram. You can see that the metabolism is redirected to make glucose and ketone bodies to supply the starving body. To terminate the signal, the cell has specific enzymes such as beta-ARC that are capable of phosphorylating the receptor. This phosphorylation then attracts a second kind of enzyme that comes in and binds to the receptor to stop further signaling. One common example for this type of enzyme is beta-arrestin. Alright, back to this diagram, let's cover the last hormone, epinephrine, which is also known as adrenaline. To do so, we need to consider the kidneys and the adrenal glands in more detail. If we take a cross-section of the adrenal glands, it can generally be divided into two main regions: the cortex, which corresponds to the outer part, and the medulla, located within. The cortex can be divided into three further zones, with each being responsible for the production and release of a given set of hormones. In our case, epinephrine is produced and released in the bloodstream from cells in the medulla. In comparison to glucagon and insulin, epinephrine is a small molecule derived from the amino acid tyrosine. The main signal that causes its release is activation of the sympathetic nervous system, which can be triggered from stress and exercise. Now, if we consider the effects of epinephrine on its target cells, the hormone impacts all three tissues, muscles, liver, and adipocytes. As you may guess from the fact that it's released in conditions of stress, epinephrine's main goal in terms of metabolism is to mobilize nutrients to get the body ready to overcome the stress. To do so, epinephrine operates on two main classes of receptors, the alpha and beta adrenergic receptors. The liver expresses both type of receptors, but the muscles and adipocytes predominantly respond with the beta adrenergic receptor. Both these receptors are G-protein coupled receptors, and the alpha is associated with a GQ protein, whereas the beta is associated with a GS protein, just like glucagon. As such, if we first consider the effects of the beta adrenergic receptor, the same adenylyl cyclase, CMP, and PKA pathway will be activated. Although this will upregulate the same catabolic pathways glucagon activated, so glycogen and triacylglycerol breakdown, as well as beta oxidation, there are some differences between the two hormones depending on the tissue. If we consider muscle cells, epinephrine's function is to mobilize nutrients for energy production, so instead of promoting gluconeogenesis and ketogenesis, the hormone will promote glycolysis and the degradation of acetyl-CoA to make ATP via the citric acid cycle and electron transport chain. To do so, muscle cells have a different isozyme, or in other words, a different version of PFK2 that gets activated by PKA. This allows muscles to favor glycolysis, whereas hepatocytes have a different isozyme that gets inhibited and thus promotes gluconeogenesis. Additionally, muscles lack the enzyme to make ketone bodies, so their acetyl-CoA gets processed to make energy only. When it comes to adipocytes, since their function is mostly to store fat, epinephrine signaling is mainly used on them to make them release the fatty acids that they store. So, in effect, epinephrine primarily stimulates the triacylglycerol breakdown and prevents fatty acid synthesis. Altogether, this forces adipocytes to release their fatty acids in the bloodstream such that other tissues like the muscles can continue making energy. Those fatty acids can also be used by hepatocytes to make ketone bodies. Now, in the liver, what we mentioned with glucagon stays true. The liver isozyme of PFK2 gets inhibited by PKA and thus promotes gluconeogenesis. With epinephrine now in the picture, the two hormones work together to accomplish the same goal since they activate the same pathway. In addition to this, remember that epinephrine increases the release of glucagon, so not only does epinephrine contribute to glucagon's effect in the cell, it increases its concentration in the first place. Now, when it comes to the alpha adrenergic receptor, the GQ pathway starts in a similar fashion to the GS pathway. The binding of epinephrine activates the receptor and the associated alpha subunit. Instead of activating adenylyl cyclase, the alpha QG protein activates a membrane protein called phospholipase C, or simply PLC. When active, PLC cleaves PIP2 into diacylglycerol and IP3. Diacylglycerol is responsible for activating protein kinase C, and IP3 is responsible for opening calcium channels in the endoplasmic reticulum. With respect to the pathways that we have here, PKC enhances glycogen breakdown by further inhibiting glycogen synthase. As a final detail on this section, I want to mention that in a similar manner to insulin, extended PKA signaling can reach the nucleus as some of its phosphorylation targets are transcriptional factors. One common example is the transcription factor CREB. Consequently, glucagon and epinephrine are also very complex hormones that have lasting effects in the cell beyond what we discussed. Nonetheless, to close this section on epinephrine and glucagon, let's recap how all the different tissues interact with each other to make sure this is clear. Under the influence of epinephrine, adipocytes release fatty acids into the bloodstream to feed muscles and the liver. In the liver, the combination of epinephrine and glucagon promotes the organ to use its nutrients to remake glucose and ketone bodies and release them into the blood to feed muscles and the brain. Muscles react to epinephrine by upregulating all the catabolic pathways and thus use the nutrients they already have and those that they receive from the blood to make energy. Similarly, the brain uses glucose and ketone bodies to make energy. Hopefully, this can clarify what each tissue does in these conditions. With this being said, when we compare all three hormones to each other, this is how they influence the pathways. Insulin promotes the storage of nutrients after a meal, and thus shunts all the nutrients towards an anabolic fate. On the other hand, glucagon is released in times of starvation and exercise and promotes gluconeogenesis and ketogenesis in the liver. These two anabolic processes are fueled either directly or indirectly by the catabolism of fatty acids. Finally, epinephrine is released during stress and exercise and promotes mainly the catabolism of nutrients in muscles and adipose tissues. In the liver, epinephrine amplifies the effects of glucagon. Alright, this final topic on hormonal integration completes our discussion on fat and carbohydrate metabolism. As I mentioned a couple of times, there is a lot that remains to be discussed with respect to metabolism, such as the metabolism of amino acids, nucleic acids, other lipids like cholesterol, and the integration of all those nutrients in the body. Thankfully, this video has allowed me to introduce a bunch of concepts that will be able to be reinvested in those future discussions and make explanations more straightforward and unified. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.